I am Eva Redei. I am somebody who has come a long way from completely different disciplines, such as engineering and organic chemistry, things that you probably hate. And I came this long to try to understand and solve a problem that is extremely serious. Well, the title says it. Depression is a disease, and it can be cured, but we don't know yet how to do it. I would like to thank all the patients, all the collaborators, students, donors, volunteers, and of course, as it was called by a journalist very promptly, very accurately, my unlikely allies, the rats that you heard about. So depression is a disease, and it is for real. You can't pull yourself together without help most of the time. You can't get over it as easily as many other people say you can. And you can just snap out of it as it's told by others to you or to people you love. 1 in 10 people of the US have major depression. It is not only the disease of the Western world or the middle class. No, depression affects everybody. Andrew Solomon, who is a writer and who himself also suffered and suffers from depression, says that depression is a family secret that everyone has. And it's probably true, and whether you know it or not, somebody in your family, among your friends, acquaintances, is suffering from depression right now. It is also an ancient illness. Hippocrates constructed the word melancholia, which we still use as one subtype of depression. And then it goes on to Galen. There is still a college in Oxford named after him, from Turkey, Avicenna from Persia, that has as close of a definition of depressive type of mood disorder as it comes for melancholia, all the way to Kraepelin, who was actually the first who described the disease to be biological and genetic as a component. And then, of course, Darwin, which may come as a surprise for many people, but actually he wrote the expression of the emotions in men and animals that describe clearly the possibility or actually the evidence that animals show expression of emotions and can show depressive expressions as well. And then finally, Freud, who is the founder of psychoanalysis, and I'm going to make a statement that is probably not very wise of me, but I do think that Sigmund Freud with psychoanalysis delayed the development of psychiatry, according to many people, to 50 to 100 years. It's extremely intellectual exercise, but it's not based in brain science. Okay, so here comes a quiz. Who are these people? Who knows? Give me just names, some names. Churchill, good. So Churchill, okay, let's just take one at a time. Churchill, Winston Churchill, of course you know who he is, and he called his depression my black dog. He was bipolar, he had bipolar disease. Okay, next. Yes? Abraham Lincoln, yes. Abraham Lincoln, our 16th president, and he has fought with depression in his whole life. Next. Oh, Van Gogh, yes, Van Gogh. I mean, officially he was accident by gone, but it was, right. Okay, more. Oh, there are some more difficult ones, haha. So more. Sylvia Plath, very good. Emily Dickinson, correct. Emily Dickinson, that's correct. She was also depressed in her entire life. Okay, so the ones that we didn't get is John Adams, the second president of the United States who has suffered from depression on and off. Ernest Hemingway, I can't see it very well from, who of course killed himself. We know that. And he had suffered both from depression and alcoholism, just like Winston Churchill, who drank 1 bottle of whiskey a day. So, okay, now that's an interesting one. Isaac Newton, who had chronic depression in his entire life. Who is this one? You come on, guys. Right? No, it's not Armstrong. Aldrin, right. And Aldrin did have major depression and suffered from it too. It's also, obviously, as I said, it's everywhere in the world. And I really wanted to show you this one as opposed to some other slides that shows depression rates around the world. First of all, you see it's close to 10%, but the interesting thing is that, sorry, it's here, United Kingdom happens to be over there. The reason I want to show you this one, because it puts Japan into the Middle East, and it puts Italy somewhere to Southeast Asia, but the bottom line is still important, which is, it's still important because it is everywhere. The disease is everywhere. Please remember some of these numbers because I am going to refer to them in the future. Okay, so it's not only the past. Here comes the next quiz, and then I put them together. Again, okay, names. Marilyn Monroe. Robin Williams. But both of them have died of, yes. Woody Allen, he's still alive and he's still depressed, yes. Anne Hathaway, correct. Come on, guys, you know her. Lady Gaga, right? Okay, you know her. Angelina Jolie, who has been depressed during her teens and early 20s, Gwyneth Paltrow, Gwyneth, I can't pronounce W's, right? So you know that all these people who are celebrities, but they were brave enough to say that they did. Now, who are these? The Dementors. And they were actually created by J.K. Rowling, who herself, she created them after her depression. Blind, right? They suck out happiness from their victims and replace happiness with the most awful memories that anybody can imagine. And the victims are left hopeless and helpless, just like people with major depression. People with depression cannot enjoy everyday things, cannot enjoy food, company. They have difficulty to get out of bed. They usually have either weight loss or weight gain. They cannot sleep or they sleep too much. These are all symptoms of major depression. So obviously, it's a very serious illness. And as a very serious illness, it needs to be treated. But before it can be treated, it first needs to be diagnosed. And that's where the difficulty starts. 30 to 40% of people do not seek help because of stigma. So what comes to your mind when you hear stigma? Yes. Perception, right. So the perception starts with, very general idea. What do you say to somebody, your classmate, who is sort of weird? First of all, you say weird. What is the next thing you say? Oh, it's a behavior. What is that? Crazy behavior. You put a qualifier, so you put things on people regardless what it is, but that implies that there is something different about them. But what is the reality? It's in a survey, it shows 50% of people believe that depression is caused by how you were raised. It's because you are too weak. And they also believe that 60% actually believes that the treatment of depression is you just have to put yourself together and pull yourself up by your bootstraps and so on and so on. And it's such a huge issue that that amount of depressed patients. So think about the 10% that is identified as have depression in the US, and I show you that's actually what is the actual percentage. So those that go to see a doctor, half of them go to their primary care physicians, and 50% of them are not diagnosed. And even that those that are diagnosed, 1/4 of them get the wrong diagnosis. So if you calculate it all together and you assume that the 50% that is diagnosed by a mental health professional is perfectly diagnosed, that is only the percentage that is diagnosed correctly. What is the result? The result is that the diagnosis and treatment is delayed for 6 weeks to 60 months. And during that time, the patient, their families, everybody around them suffer. We need better diagnosis. And better diagnosis starts with biomarkers. So what is a biomarker? Biomarkers are, of a very specific illness, something that objectively you can measure objectively. And you can measure objectively as an indicator of a biological process, a pathogenic process, or an illness, basically, or a pharmacological response to therapeutic intervention. This is a very complex official definition, but biomarker is actually something that reflects the disease, either reflects it how it is now, or prognostic, it says how it is going to be, or what treatment is it going to be useful. Now this is, just bear with me because this is a very complex one and I don't know whether you can even see it. So biomarkers can be at any, and right now I'm talking about biological, blood-based biomarkers basically. So they can be anywhere on this path, which is the genome, the epigenome, and that Barry talked about, the transcriptome, the proteome, and the metabolome. What does it mean? Genome is your DNA. Every cell has the same DNA, but not every cell does the same thing. And of course, because at the top of the DNA is modified by very specific modifiers so that the different cells can do different things at different times also. And so it's usually this modified epigenome is reflected in the transcriptome. It's expressed. So DNA is the genetic code, and it's the message, the copy of the DNA is the RNA, and all of the RNA is the transcriptome. And then that sends the message, the genetic message from the RNA to the protein machinery, whereby the cells can make proteins, okay? And certain type of proteins regulate your metabolism, and that's basically what's called a metabolome. So if you think of it, every one of these could be a biomarker. Well, we have a problem for depression. Until recently, these huge genome-wide association studies, you learn what they mean, had no positive outcome. Didn't find any significant sequence variation that is associated with major depression. Very recently, there was a study that looked at only women, only chronically depressed, very severely depressed women, and they found 2 sequence variations. So now we could even try to do this, but depression is a very heterogeneous disease. We don't have much of this yet, and nevertheless, I decided to focus on the transcriptome. Basically, look at RNA. And so what we want to discover is we want to discover a laboratory blood-based test that objectively and precisely and timely can diagnose depression. Objectively, because the current way of diagnosing depression, do you know what it is? Somebody sits down with you and there is a clinical interview while you are answering to all the questions. And if you are not willing to or not able to answer those questions, here goes the precise diagnosis. Precise because you saw the objective diagnosis. Precise because you saw that many of the patients are misdiagnosed, and timely because it's instead of 6 weeks to 60 months, the test that I'm going to tell you shortly can be done in 2 days. Okay, we also aim to do a blood test that predict the efficacy of treatment, which will help each patient. At the moment, the treatment is, there's a trial and error treatment. So people come in, they get the first antidepressants or the first psychotherapy. If it doesn't work, then they get an adjunct therapy or they move to another one, and each of them to 6 to 8 weeks. And because only 40% of the patients respond to antidepressants, that means a long time for that 60%, even if they respond at all. And then finally, a large percentage of patients have recurrent episodes. One episode, quiet, one episode, quiet. And those that have these recurrent episodes are much more ill than if they would have only one episode, obviously. They can get maintenance therapy, either psychotherapy or blood treatment. So we chose to look at RNA because it's easy to collect from blood and transport. There are multiple RNAs in the transcriptome, not only those that will be translated into proteins, so we can capture a larger variety. And there is an easy analysis, well-known system called quantitative reverse transcriptase, PCR, or others that are currently available. Even though I said transcriptome, there are many ways to do this. You can use only human samples. You can identify candidate biomarkers and test them. Or you can test a panel of markers used for multiple purposes. Nevertheless, we used this, which is a genome-wide expression in an animal model that followed by validation in humans. Here is a valid animal model. So this is our strategy. But they have to share common phenotype. So what we did is we discovered biomarkers in the animal model, which is rat, and then test them in humans. Now, how do we define animal models being, how can we see that these animals are depressed? So here is what I told you. This is the criteria for major depression today. You have to have one of these, either feeling helpless and hopeless, or have loss of interest or pleasure, plus any 4 of these symptoms. That is the current criteria for somebody to be diagnosed with major depression. So how we can test an animal that it's feeling helpless or hopeless? So if you put them into an environment that is not a very pleasant one, normal animals are trying to escape. The animals that are showing this phenotype of depression phenotype, just like humans, give up. They don't try to escape. They become helpless. Loss of interest or pleasure, they are not interested in food or sex. It's extremely difficult to breed them. But nevertheless, it's crystal clear if you put some food in front of them that others really like, they're not interested. They have loss of motivation. They don't go for a treat. They are actually much lighter than their genetic counterparts. They are very close. They have difficulty to sleep. They have lower, their REM sleep, which is a part of the sleep pattern, is different. And so these are all can be measured by tests. How did we use this? We know that depression is about 38% genetic, and the rest is environmental. So we had decided to have 2 models. One of the models is the genetically the genetic model of depression I just described. And the other model is an environmental stress model. You can stress an animal with a psychological stressor that doesn't do any harm to them other than they become stressed and they show symptoms of behavior. And then what did we do? We have taken the blood and brain transcriptome. Remember, I'm just reminding you, what does it mean? It's all the RNA. It's actually more than mRNA. We took it from both models. We compared the depressed model with its control and the stress model with its control, and we got the differences in their transcriptomes. And what we saw that they are different. They did not overlap. So you have the depression that's genetic and the depression that's environmentally induced has different pattern, different networks, different pathways. Nevertheless, we took those that overlap. So one of the criteria is depression is not in the blood, it's in the brain, right? So that was a major hassle that we had to overcome. But it's been shown that in reality, if you take blood, they take any other tissue, not only brain, from the same person and blood, and you compare them, there is a major overlap in their transcriptome, meaning that is also a signature of the person. So we did the same. We took the 3 different brain regions, the frontal cortex, amygdala, and hippocampus of the same rats, and also their blood. And we looked the differences in the amygdala between the depressed and the non-depressed rats, the differences in the hippocampus, the differences in the frontal cortex, and the differences in the blood. And what overlapped between the blood and the different brain regions, we defined that those are markers because they reflect in the blood what's happening in the brain. And they reflect what's happening in the brain of the depressed animals. So now we took these two sets of markers from the genetic model and the stress model. We combined them since there was no overlap, and we tested them in adolescents. So these were 15 to 19-year-olds. Many of you fall into this category. I don't know whether you're aware of it, but that is an extremely vulnerable age. From 11 to 17, the rate of depression rises from 5% to almost 12% of the population. That is difficult, and you probably know it right now than how I remember it. That was a long time ago for me. So it is a difficult stage of everybody's life, and it's also difficult to diagnose. So what we did is we took these biomarkers that we translated into human transcripts, and we measured in the blood of these adolescents and compared it to adolescents that matched by sex and age. And what we found that an 11 marker panel could separate who was depressed, who was not depressed. The depression was determined by a psychiatrist, and our lab did the analysis blind. So these marker panels were able to distinguish depression with high precision, and it was greeted with a lot of press. And a lot of press is not because of us, it's because what it means for people, what it means for the young ones is decreasing the stigma. If something is in your blood, you can measure it. It's not something that you're imagining. It's right there, just like diabetes is right there. It's not your weakness. And same thing, it was really great, it was really the press and everything gave major attention to it. Again, not because of what we did, but because of what it meant for everybody. So then we moved to adults, and now this was an absolutely clinical study. We had adults aged from 23 to 83 that some of them had medication but were still depressed. Others never had medications, but everybody was still depressed. Then they received an 18 weeks cognitive behavioral therapy, which is a psychotherapy that basically trying to change your negative thinking into a positive one. And so we took blood before the therapy and after the therapy. And what we got, that 9 of the panel could crystal clearly differentiate who was depressed, who were not depressed, regardless whether they had antidepressant, whether their age, whether their sex. They said these people are depressed. Then we could also determine who will likely get benefit from the cognitive behavioral therapy, because not everybody did. Only about 40% of the patients who were there, who participated, got completely well. And we could determine from the pattern, from the network that Barry was referring to, the pattern of the co-expression of these markers, who will benefit from psychotherapy. So again, we were very lucky because the interest that it received, by the way, I had no idea what Mashable is until somebody sent me this, and I'm not so sure I still know, but you do probably. So again, emphasized, not again what we did, but how important it is to move this forward. So what is it that patients want? They say it, they send emails, they call up. They want an objective diagnosis without a stigma, because that would eliminate the stigma. And it also eliminates the negative consequences. If it's on their record that your test is positive for depression, sooner or later, it will have the same meaning that you have a blood test that says you have high cholesterol. Sooner or later, not immediately, but it will. They want effective treatment that works fast. They want to know what is that effective treatment. They want to know if they have another episode that is likely. And they want to return to normal. I'm just trying to emphasize there is another disease that is as prevalent, although this is real. This is probably not, because I said the diagnosis is very, very subjective. That has the same prevalence rate. Depression costs more per year, and still the spending on depression research is only 1/3 of what's spent on diabetes. And so finally, what is the future? The future is what I just repeat, I repeat. Objective diagnosis. It decreases the treatment delay and also decreases stigma. Predicting what treatment an individual should get, and it would have adherence to treatment better and a much greater rate of recovery. Treatment decisions, measuring how efficient a treatment is, and finally, what's really the next is whether these biomarkers represent a consequence of the disease or they are participate in the development of the disease. Because if they do participate, then they represent novel drug targets for depression. And the last drug that was developed based on an entirely non-bioamine-based theory of depression was never. We only have a family of antidepressants that are based on one theory. And that needs to change because there is very limited efficacy of them. They're good when they work, but not all the time. That's it. Thank you.
70% of people becoming depressed is because of the environment and stress. This was one of your first.
Yeah, it's 60. It's 60%.
And can
right.
you detect that with the biomarker, or there is no difference between genetic and environment?
Right. We can't, because the genetics of depression is not really elucidated, and it's so heterogeneous. When we collect, the depressed patient come because they are depressed. We don't know whether it's an environmental cause or a genetic cause. So we detect, this combined biomarker can detect, but we don't know which one. Yes?
Thank you. You had said that the blood test predicts who is depressed 9 out of 10, and you also said that it also predicts who's going to benefit from psychotherapy. Is that like two different things? So the people who are depressed, some of them have a different, are you saying different forms, or that different types, because one will benefit and one won't? Or was there any correlation with that?
Right. So we don't know whether they are different forms, but based on the biomarker, the probability that is that they are. But again, just you have to understand, depression is very heterogeneous, right? So, and we still have these 9 markers that can detect them all, which means that this is just the top of the tip of the iceberg. So it's more and more and more will come. But anyway, we are very happy that we have some right now. Yeah.
So you discussed how you would find traits of depression in rats. My question, and how you were saying, as they're depressed, they're not interested in really anything, and how difficult it is to find them. How do you get a large enough data pool, if you will, or how do you get enough rats that are depressed to get this much information about all their depression?
So we've been breeding these animals very somewhat similarly that was described for alcohol consumption. We've been breeding them for 35 generations. I've been working on this project for 17 years. So the information didn't come overnight. We bred them we mated them as much as we had to in order to maintain the colony.
Thank you. Thank you very much.